So my article was on gene therapy, called Gene Therapy Second Act. So basically the definition of gene therapy is the transfer of genetic material to cells or tissues to prevent or cure disease, and just basically taking healthy genes and replacing those defective or diseased genes. It's used to cure hereditary diseases such as like single cell polygenic. For example, like single cell diseases are like muscular dystrophy and hemophilia, which is a type of blood cancer. It's also used to cure monogenic disease or multifactorial diseases. But the problem with like multifactorial diseases is there's different factors that come into effect besides genetic factors. There's also environmental factors. So like diabetes, for example, can be genetic, but then you can also obtain it as well. So there's steps in the gene therapy process. One, deciding how to cure someone with it. So first is choosing the gene that's need to be used to perhaps produce a protein that person is lacking. Then deciding which vector to use. So I will discuss it later, but there's two types of vectors, viral and both non-viral. And then also targeting the certain cells using promoters, which go to different types of tissues, and then also ubiquitous promoters, which target multiple tissues at once. And deciding the route of administration by either developing the DNA and then injecting it directly in vivo into muscle or cranial, or developing ex vivo and then injecting it into the patient. So there's two vector types in the types of administration, both viral and non-viral. In viral, you have 4 different types of viruses that are adaptive to inject the genes. Those are retroviral, lentiviral, adenoviral, and adeno-associated. And then in non-viral, use plasmids. So this is just a visual before I start discussing the different types of vectors of how exactly they do it. And basically it just shows here taking the virus, the viral package, and stripping it of some of its DNA and inputting the therapeutic gene, which then goes into the cell and then starts developing the therapeutic proteins that the person is perhaps missing, and then to cure the hemophilia or the muscular dystrophy. So retroviral and lentiviral are both derived from these similar viruses. They, after getting stripped of their DNA and then putting the therapeutic gene and then injecting into the patient, they will enter the cell, and the RNA genome will then reverse transcribe and then make the DNA that will then enter the nucleus to start developing the protein. So once, for example, retroviral, once it enters the cell and starts developing the protein, the cell divides, and then the body starts mass producing that protein. Although it's only limited to ex vivo because it cannot enter single cell and non-dividing cells, it can only enter quiescent cells. Lentiviral works both in ex vivo and ex vivo, so it can enter both dividing cells and single non-dividing cells. And they both have a capacity of 8 kilobases, which basically means kilobases are the pairs of DNA that the viruses can contain, or the DNA can contain, and those are both at like a medium capacity compared to the other two. So genoviral has a double-stranded genome and uses a different process of entering the cell once injected. It uses a process called endocytosis, and it enters the cell as an endosome, and then after transcribing the DNA, then enters the nucleus and starts producing the protein. The affected cells are both dividing cells and single cells, so it works in both in vivo and ex vivo. There's also something called a gutless adenoviral vector, which basically means they take an adenoviral virus and strip it completely of its viral genes and then inject the therapeutic gene, basically utilizing maximum capacity, which also shows that it has the most capacity of 37 kilobases of all the viruses. So adeno-associated vectors have a single-stranded DNA, and they also use a similar process of endocytosis and enter the cell as an endosome. After entering the cell, it becomes a double-stranded molecule, and then injects the DNA into the nucleus of the cell and starts producing the protein. It also affects both single non-dividing cells and dividing cells, and due to its structure, it's able to maintain itself well in the cells, which is why it can be maintained for years. although the only limitation it has is that it only can contain up to 4.5 kilobases of like, DNA space. So for non-viral vectors, they use plasmids, and plasmids are better because they have no size limitation, and because they are non-viral, the body doesn't have any immune system reaction to the vector once injected. Although they transfer directly into the tissues, they don't have as efficient of a process of entering the cells as do the viruses because they're not as infectious. But to increase the permeability of these vectors, they use physical methods such as electrotransference, which just uses electric pulses to make the plasmids more permeable, and then also sonar operation, which uses sound waves. And they also have chemical methods, which are like lipoplexes, which is a complex of plasmids and lipids, and basically encases the plasmid and then allows it to be more permeable. Same with polyplex, it's a complex of polymers. So basically the article mostly talked about was setbacks and breakthroughs in gene therapy. So a couple of setbacks they had was Jesse Gelsinger had an immune system reaction to gene therapy. He was treated for, I believe it was hemophilia, and then his liver had an adverse reaction to the gene therapy, which actually proved fatal to him. There's also some children that had severe combined immunodeficiency X1, and when went through the gene therapy process to try and cure that, 5 of the 20 children ended up developing a form of leukemia. So although there were some setbacks or more breakthroughs, for example, gene therapy restored sight to 40 patients who had a hereditary form of blindness. 120 plus patients with various blood cancers were also cured, and some of which are now free of malignancy for over 3 years. Corey House had a form of degenerative sight where his sight over time just kept decreasing, and then once after gene therapy, he eventually came back, and now he's well enough to go turkey hunting with his father. Then there's a treatment called chimeric antigen receptor, which basically uses T cells as the vectors, and then those T cells, which are a part of your immune system, target the cells that carry leukemia, which is how I have a video on Emily Whitehead was a success story. Is that right? Just open it.
Yeah. It has amazing its own sometimes. Oh, my computer is on mute. On the top keys, on the F keys, there should be one that is red. Just click it.
Unless it would come from there. Hold on.
There's no audio on this
This should be the speakers.
Is there an audio cord?
No, but the audio for there is, but
Then I can see the DJ.
yeah, PG.
Oh, okay.
Family turned to the experimental therapy at the Children's Hospital in Philadelphia. Dr. Stephen Crump is leading the study.
Yeah, that all right.
Just switch your mouse on.
Yeah.
Or small.
Well, you can hear that all right.
Can you hear it?
Yeah.
I think the phone have to come.
Yeah, we can hear that.
Okay.
We have some promising news to share tonight in the fight against leukemia. An experimental gene therapy is continuing to show high rates of success in patients who have failed to respond to traditional treatments. We first met Emily Whitehead a year ago. She was 7 then, battling ALL, acute lymphoblastic leukemia. When she didn't respond to chemo, her family turned to the experimental therapy at the Children's Hospital in Philadelphia. Dr. Stephen Crump is leading the study. This is a whole new way of treating cancer. The idea is that we're study have experienced complete remission.
So that's just one example of what gene therapy has done for patients. And I have one more short video that just basically explains what gene therapy is, somewhat more of like in layman's terms, I should say.
Gene therapy has the potential to save millions of lives if we can just figure out how to make it work. Hey peeps, thanks for tuning in to Gene Therapy sounds like
What do I have to do?
Hit escape. Get out of the problem. And switch the screens. Stand it again. Sorry, I use this with my monitor.
Here you go, sir. Thank you.
nice easy treatment, right? It's therapy. In some ways it is, on the macro level, but in your cells it's a little bit invasive. In gene therapy, doctors are basically hacking the DNA of a living human. Using genetically engineered retroviruses called vectors, scientists infect human cells. The retrovirus can be programmed to carry a gene or a bit of DNA that will overwrite the messed up mutation and make it work properly. It was first tried on a young girl in 1990, and despite some early failures, it has the potential to revolutionize treatment of genetic disorders. The journal Science describes one of the recent successes that gene therapists say was really exciting. A few children were born with metachromatic leukodystrophy, which causes a defective immune system and some brain disorders, and kids who have it usually don't live past the age of 5. Bone marrow contains stem cells. The cells normally produce red blood cells, but they can be reprogrammed using gene therapy. It's a little risky, but it can work. Taking bone marrow from these kids, doctors were able to infect the cells with a retrovirus and replace the stem cell's mutated gene with a repaired gene. Then they re-injected that back into the patient, and the fixed cells multiplied, and as of the time we filmed this, the patients are all in good condition and are heading to kindergarten at the time that others with that disease can't even speak. There may be future side effects, but they seem pretty happy with the result at the moment. I mean, I would be. Science just helped some kids. Woo! It's not just useful in children. Scientists have also used gene therapy on dogs to cure them of type 1 diabetes, with 2 of their doggy patients still alive years later. The treatment involved injecting 2 things into dogs' muscles, one gene to sense glucose and an enzyme to dictate glucose absorption. Scientists don't have to target our DNA. They can also use gene therapy to target the DNA of cancer cells. It's like they gave cancer cancer. You've seen this before if you've been following D-News. A protein called CD47 is like a passport that tells your immune system not to attack a cell. Normally, cancer produces a ton of CD47. Using gene therapy on the cancer, scientists turned off that cell production and let the immune system blow it out of the sky like a decloaked Klingon bird of prey. Gene therapy is still in its infancy, but the promise of future cures for everything from cancer to genetic disorders is pretty incredible. And I, for one, cannot wait to see what this brings. What do you think of gene therapy? How do you see it being used in the future?
And that's it. These are my references from where I got the article, and then also another extra video that I watched to get a little more information about gene therapy.